The overall goal of this procedure is to introduce both lysine and arginine-type monomers within the same peptoid sequence, and to evaluate the activity of these compounds against Leishmania mexicana, the causative parasites of the neglected tropical disease cutaneous leishmaniasis.
Novel therapeutics are urgently needed for cutaneous leishmaniasis, and this procedure can help to identify and develop peptoids as a new class of potential anti-infectives for this neglected tropical disease. The main advantage of this protocol is that peptoids can be synthesized that contain mixed cationic functionality. This is highly desirable as it can help to modulate the biological activities of the sequences. Although this tests against the mammalian form of the parasite, it can also be used against the insect stage of Leishmania mexicana or applied to mammalian cell lines. Visual demonstration of this method is really useful because it shows the stepwise nature of synthesis and also the parasite culture at various points in the life cycle of Leishmania mexicana.
To begin this procedure, prepare solutions in dimethylformamide as outlined in the text protocol. Then add 0.1 millimoles of FMOC-protected rink amide resin to a capped 20 milliliter polypropylene reaction vessel with 2 frits. Add 5 milliliters of dimethylformamide and let the vessel stand for 60 minutes at room temperature. Then, drain the DMF using a solid phase extraction vacuum platform. Next, add 2 milliliters of prepared piperidine solution. Place the vessel on a shaker platform at 450 RPM at room temperature and shake for 5 minutes.